Alright, back to this diagram, we have covered glycolysis of all the important sugars, the regulation of the pathway, and homolactic fermentation. What I want to discuss now is how some specialized cells in the body can take some pyruvate and reform glucose in a pathway called gluconeogenesis, which is essentially glycolysis in reverse. I want to mention right away that just like we did for the previous pathways, I will focus on the reactions and metabolic principles, but when it comes to the physiological integration, that will come later. I can already tell you that the liver is the primary organ that performs gluconeogenesis, but again, the details for how that is mediated will be later. Now, the reason why I say gluconeogenesis is essentially glycolysis in reverse is because when we consider the reactions of this pathway, you can see that it shares most of the steps of glycolysis, with the exception of a few that are highlighted here and on the map. To understand why some of the steps differ, we need to consider back the thermodynamics of glycolysis and discuss a concept known as substrate cycling. As you can see from this table that we have seen a couple of times now, we have the changes in free energy associated with each reaction. Based on these values, remember that reactions with very negative values, such as those highlighted in violet, correspond to irreversible reactions, and those with values close to zero correspond to reversible reactions. In the cell, what this means essentially is that irreversible reactions will flow to make products regardless of the concentrations, whereas reversible reactions will flow to equilibrate both products and reactants. To illustrate this idea of why it can potentially be an issue for cells, let's consider this small pathway of A producing F through various intermediate steps, with one being irreversible. Let's also consider a hypothetical plot of substrate quantity along the progression of the pathway. So, if we imagine that we start with some amount of A, this amount will equilibrate between A, B, and C, as all of the reactions linking them are reversible. If we imagine that this given amount of C reacts to completion to produce D, then the quantity of D will equilibrate between E and F to make our final product. I stopped the pathway here, but obviously A would keep on equilibrating with B and C, and so on until everything is balanced out, but this aspect is not the concern for us. The issue with a pathway like this arises when we consider what happens when we start with a large amount of F, or in other words, the final product. As you can expect, this amount of F will equilibrate with E and D, but it will get stuck there because there is no amount of D that will be able to overcome the irreversible reaction between C and D. As such, if F accumulates, then so will E and so will D. In a physiological setting, this accumulation is likely to become toxic for cells and the body. To circumvent this issue, cells generally have a reverse irreversible reaction that will allow to go back in the other direction. This set of opposing reaction is essentially what substrate cycling is, and as you can see, it allows the pathways to operate in either direction. Now, if we consider A to F to instead be some substrates of glycolysis and gluconeogenesis, hopefully you can see why the two pathways share all the reversible steps but differ on the irreversible reactions. I want to point out that if we strictly consider the pathway at face value, you might be wondering what will happen when we get to the reaction between C and D. Will it go towards A or will it go towards F, given that the two reactions are irreversible? It turns out that the directionality can be controlled by hormones, and as we will see later, if a given hormone has the effect of stimulating or inhibiting one direction of the pathway, it will cause the opposite effect in the other direction and thus lead the substrates to either go towards A or F. We will come back to this aspect of hormonal control to add more details on how this occurs. Now that we know why gluconeogenesis and glycolysis are organized that way, I want to take a look at the reactions of gluconeogenesis. Given that this pathway only differs from glycolysis at 3 instances, which we now know are 3 substrate cycles, I'll focus on these different reactions, but keep in mind that we have seen all the other reversible reactions when discussing glycolysis. So, starting from the last reaction of glycolysis, which is the formation of pyruvate from PEP, the aim of the first reaction of gluconeogenesis is to reform PEP. Now, although gluconeogenesis is typically considered the inverse of glycolysis, pyruvate is not the actual starting point for this pathway, but rather it is oxaloacetate. Oxaloacetate is a substrate generally associated with the mitochondrion as it is part of the citric acid cycle, which we will cover in the later section. What is important to understand for us at the moment is that oxaloacetate constitutes a good starting point as many substrates can tie back to it. For example, pyruvate, many amino acids, and other citric acid cycle intermediates. With respect to pyruvate, the reaction that converts it to oxaloacetate is catalyzed by pyruvate carboxylase, which adds a CO2 or carboxyl group to pyruvate. You will notice that this reaction requires some energy in the form of ATP hydrolysis, so producing 2 oxaloacetate molecules from pyruvate costs 2 ATPs. A crucial detail about this reaction to mention, when we consider how it is integrated in the cell, is that it only happens in the mitochondrion. This is a little problematic because the rest of gluconeogenesis happens in the cytosol. Thankfully, cells have a mechanism to fix this called the malate aspartate shuttle, which allows to reform oxaloacetate in the cytosol. We will cover the malate aspartate shuttle in more detail later when discussing the reactions of the mitochondrion, but for the moment, just keep in mind that this shuttle allows oxaloacetate to reintegrate the gluconeogenesis pathway. Now, with the oxaloacetate present in the cytosol, the enzyme PEP carboxykinase, or simply PepCK, uses a phosphate group from GTP to replace a carboxyl group of oxaloacetate and thus reform PEP. Since the CO2 or carboxyl group is removed from oxaloacetate, this reaction is called a decarboxylation, and it turns out that it releases the sufficient amount of energy to reform PEP. As such, oxaloacetate is also referred sometimes as a high-energy intermediate, and it is because of this reaction. As a quick note, even though GTP is used in this reaction instead of ATP, the two molecules are energetically equivalent, so in total, 4 ATP-like molecules are used to synthesize 2 PEPs. Before we move on to the next reaction, there are 2 aspects about this specific step that I want to point out. First, you will recall from our discussion on homolactic fermentation that pyruvate can interchangeably be converted to lactate, and as such, lactate also constitutes a potential starting point for gluconeogenesis. The second aspect, which you might remember from our conversation on glycolysis regulation, is that pyruvate kinase can be inhibited by the action of an hormone called glucagon. Glucagon is a hormone that typically gets released under fasted conditions and signals to the liver to release glucose in the blood to maintain appropriate levels. One way to obtain this glucose in the liver is obviously to perform gluconeogenesis, so in other words, glucagon stimulates gluconeogenesis. As such, to make sure PEP is used to reform glucose, glucagon inhibits pyruvate kinase and thus prevents wasting away PEP to make pyruvate. This regulation illustrates very well the idea of substrate cycling, as a hormone can directly dictate which pathway the cell uses. This idea of substrate cycling and hormonal control is even further exemplified in the next step of gluconeogenesis, which occurs between fructose 1,6-bisphosphate and fructose 6-phosphate. Recall that in glycolysis, F6P was phosphorylated by PFK1 to yield F16P. In gluconeogenesis, the opposite reaction is mediated by fructose bisphosphatase 1, or simply FBPase 1, and leads to the dephosphorylation of F16P to get back F6P. As we discussed a little when we covered substrate cycling, here we have 2 irreversible steps that look seemingly futile as F6P produces F16P and vice versa. To give this pathway some direction, here again, hormonal control will be important. Besides hormones, which we will get to shortly, an important compound that regulates glycolysis and gluconeogenesis is ATP itself. If we first consider low energy conditions, which are those when ATP stores are exhausted, an important signal to indicate low energy is the presence of AMP. If you recall our conversation on glycolysis, you may remember that AMP is a very potent activator of PFK1, but it turns out that it's also a very potent inhibitor of FBPase1. So, under low energy conditions, when the stores of ATP are depleted and AMP is high, glycolysis will be promoted. This makes perfect sense that this happens under these conditions, as one of the primary purposes of glycolysis is obviously to produce energy. On the other hand, if energy levels are high and ATP stores are replenished, ATP at high concentrations acts as an inhibitor on PFK1. Consequently, with less PFK1 activity, gluconeogenesis happens. This also makes sense because, as we've seen in the previous step, with pyruvate carboxylase and PepsiK, gluconeogenesis requires an input of energy to kick off, so having high amounts of energy allows this process to happen. Now, when it comes to the hormonal control of this substrate cycle, everything happens at the level of fructose 2,6-bisphosphate. Again, if you recall our discussion on glycolysis regulation, F26P was a key regulatory molecule in this pathway as it was a potent stimulator of PFK1. In a similar fashion to AMP, F26P has dual effects on the substrate cycle, as it is also a potent inhibitor of FBPase1. As such, the quantity of F26P plays a central role in governing the direction in which the pathway goes. This given quantity of F26P is equilibrated by the activity of PFK2, which produces F26P from F6P, and FBPase2 that degrades F26P to remake F6P. With respect to hormonal regulation, a very important point to know about PFK2 and FBPase2 is that the two are actually part of the same enzyme, and as such, hormones that stimulate one of the domains will also influence the activity of the other domain. For example, if we consider glucagon first, this hormone, which as we said is responsible for increasing glucose levels in the blood, inhibits the PFK2 domain and thus reduces the quantity of F26P. With less F26P, the inhibition of FBPase1 is released and the stimulation of PFK1 decreases, altogether resulting in the promotion of gluconeogenesis. On the other hand, the hormones insulin and epinephrine have the opposite effect as they both stimulate the PFK2 domain and thus increase the quantity of F26P. This increase subsequently promotes glycolysis. Although they have the same effect on this pathway, insulin and epinephrine are two hormones used in very different contexts. Insulin is released after meals and promotes glycolysis to decrease blood glucose levels, whereas epinephrine promotes glycolysis in muscles to make energy to supply muscle contractions. Also, as a small note, this differential regulation can be made possible because different tissues, for example the liver and the muscles, express different isozymes or simply different versions of this PFK2 FBPase2 complex. Now, one thing about this entire process that confused me a little when I first learned it is why would the cell care to make this extra F26P reaction if it doesn't even participate directly in the pathway? Why not have the hormones directly alter the activities of PFK1 and FBPase1? I think that from this model you can see pretty clearly that, for reasons of efficiency, having the hormonal control done on F26P allows to simultaneously stimulate and inhibit a given pathway, thus ensuring that the cell truly commits between glycolysis and gluconeogenesis. Finally, the last unique step of gluconeogenesis is the reaction between glucose and glucose 6-phosphate. For glycolysis, hexokinase is the enzyme responsible for phosphorylating glucose, and in gluconeogenesis, glucose 6-phosphatase is the enzyme responsible for dephosphorylating G6P to remake glucose. In this substrate cycle, the regulation to flip between gluconeogenesis and glycolysis is a little less extensive and known about, but one important aspect to mention is that the enzyme glucose 6-phosphatase is expressed specifically in the liver and the kidneys, which are the main organs performing gluconeogenesis. So, when we consider back the entire pathway, the energy balance sheet of gluconeogenesis requires 2 pyruvates, 4 ATPs, and 2 GTPs to produce 1 glucose. Recall that through glycolysis, 1 glucose molecule produced a net 2 ATPs. Before we conclude this section, I want to mention that the bulk of the energy that is provided to supply gluconeogenesis comes from the degradation of fatty acids via a process called beta oxidation, which we will cover later when discussing lipids. I want to point out also that although we have discussed many allosteric and covalent ways to regulate glycolysis and gluconeogenesis, the coordination between glycolysis and gluconeogenesis can also happen on the long term via gene expression. With some of the hormones that we have talked about, their effects can reach the nucleus, where they end up altering the amount of proteins made and thus dictating the fate of the cell even more. Alright, going back to this pathway, we have covered glycolysis of glucose and other 6-carbon sugars, homolactic fermentation, gluconeogenesis, and the regulation of all these processes. With that in mind, we are now ready to talk about glycogen, which as a molecule serves the essential purpose of storing glucose. Thank you for watching this video. If there was anything unclear or there was a mistake somewhere in the video, make sure to let me know in the comment section. If you enjoyed this video and found it useful, you can consider leaving a like and subscribing to support the channel. Thank you again for watching, and I'll see you in our next discussion.